morning, my dear students. Today we are going to learn about the biosynthesis of cholesterol. In the last video, we have learned so many things about cholesterol, its nature, its isomers, everything. So in this video, we are going to see the biosynthesis, how cholesterol is synthesizing in our body. So before that, we need to understand the significance of acetyl-CoA. This acetyl-CoA, just as the same way as a brick. What is the role of a brick in constructing a house? We know that without bricks we can construct. It is the ultimate element of a building, whatever construction. So like that way, this acetyl-CoA, it provides all carbon atoms of the cholesterol. Clear? So that is the significance. Acetyl-CoA, it is a fundamental basic unit. And next, we need to understand about two scientists, Sir John Conforth and Vladimir Krylov. What their role? They explain, they elucidate that biosynthetic pathway of cholesterol and received Nobel Prizes in 1975. And which are the sites where all this cholesterol, main sites of synthesis of cholesterol? Mainly cholesterol is synthesized in liver, adrenal cortex, testes, ovaries and intestine. Okay, so these are the main sites. Along with that, all nucleated cells can synthesize cholesterol, including arterial walls. Okay. And the enzymes involved in the cholesterol synthesis, where it is located? Partly it is located in the endoplasmic reticulum and partly in the cytoplasm. So in both cytoplasm and endoplasmic reticulum, this enzyme are located. Then we are moving to its synthesis. Its synthesis we are learning through several steps, and the first step is a condensation step. We know that condensation means combination of compounds with through the elimination of water. So here what is combining? Two molecules of acetyl-CoA. I already told you the acetyl-CoA is a basic fundamental unit. It provides all the carbon atoms of the cholesterol. So two acetyl-CoA is combining, they are condensing, and which is the enzyme. As two acetyl-CoA is combining, the enzyme also has some relation with its name of that compounds. So the enzyme is acetyl-CoA synthase. Where it is located? It is located in the cytoplasm. Cytoplasmic acetyl-CoA synthase. So what is forming? Aceto-acetyl-CoA. Why? Because two acetyl group is joining. By that way, it becomes aceto-acetyl-CoA. And the next step is the production of HMG-CoA. So this is the next step. For forming HMG-CoA, along with the aceto-acetyl-CoA, one more acetyl-CoA is combining or condensing. So that is this acetyl-CoA, it is condensing with the aceto-acetyl-CoA and forming the HMG-CoA. HMG-CoA, this one. The enzyme is HMG-CoA synthase. Which is the enzyme? HMG-CoA synthase. This HMG-CoA means what? Hydroxy methyl glutarate CoA. And along with that, we are specifying the position, beta hydroxy. In beta position, hydroxyl group. And another beta position, methyl group. So beta hydroxy, beta methyl glutarate CoA. It is forming. And these two steps, first step and second step, it is also present in the pathway of ketogenesis. So these two steps are common between cholesterol biosynthesis and ketogenesis. Okay. This HMG-CoA, it is present in both cytosol and liver mitochondria. So what's the difference in these places? That HMG-CoA that is present in the liver mitochondria, it is used for ketogenesis, and that HMG-CoA that present in cytosol, it is utilized for cholesterol synthesis. Okay, so that is the difference. Next, we are moving to the third step, It is a most committed step. It is a rate limiting step, and it's a unique step for the cholesterol biosynthesis, that is the reduction of this HMG-CoA. HMG-CoA undergoes reduction with the help of the enzyme. Need to note the name of this enzyme. HMG-CoA reduct is very, very important, and almost all regulation of cholesterol biosynthesis is occurring depending on this enzyme. So don't forget this name, HMG-CoA reductase. It undergoes, this HMG-CoA undergoes reduction and forming what? Mevalonate. Mevalonate, I will show you. Mevalonate is forming. It undergoes reduction. It receives that hydrogen from 2NADPH, and mevalonate is forming. And at this point, I already told you the step 1 and 2. It is common between ketogenesis and the cholesterol biosynthesis. So this is the ketogenesis pathway. We can see the here acetoacetyl CoA formation and HMG-CoA here also. Okay. The third step, this is the rate limiting step unique to the cholesterol biosynthesis. Next, we are moving to the fourth step, that is the production of a 5-carbon unit. Next intention is to synthesize a 5-carbon unit from the 6-carbon unit compounds. How? By undergoes a series of three steps of phosphorylation. It is happening to which compound? This mevalonate. Mevalonate undergoes phosphorylation in 3 discrete steps and helps in the formation of a 5-carbon unit. That's what we are going to see. This is the first step, phosphorylation. We know that phosphorylation is always done by some kinase enzyme, and phosphate group is receiving from ATP and forms phosphomevalonate as it BS1 phosphate group. Again another round of phosphorylation and forming pyrophosphomevalonate. Pyro means it contains 2 phosphate group. So that is formed here. It again undergoes another round of phosphorylation and will have 3 phosphate group. That is 3 phosphor, 5 pyrophosphomevalonate. So by that way 3 phosphate. Here 1 phosphate will be there. Pyro means 2 phosphate. So that is 3 phosphates get added along with the mevalonate and forms this compound. 3 phosphor, 5 pyrophosphomevalonate is formed. And this compound, it undergoes decarboxylation, that is the elimination of carbon dioxide. By that way 1 carbon is eliminated, removed. Which enzyme is helping that process? As it is a decarboxylation reaction, certainly a decarboxylase enzyme is helping that reaction and form our intended compound, that is a 5-carbon compound. What is its name? As it is a 5-carbon compound, pentenyl. It is called pentenyl. Isopentenyl pyrophosphate. Isopentenyl pyrophosphate is formed there. Okay. So all these other compounds are all 6-carbon compound, and here we got the 5-carbon compound, isopentenyl pyrophosphate. Till here, it forms the first phase of cholesterol biosynthesis. Then we are moving to the next phase or the next step. That is the condensation. Condensation of the lastly formed isopentenyl pyrophosphate. That's a 5-carbon compound. 6 such 5-carbon units, they are condensed, they are combined to form the 30-carbon compound. What's its name? Squalene is forming here. Squalene, a 30-carbon compound, and still it is a straight chain. We know that cholesterol, it has a peculiar arrangement. What's its name? Phenanthrene like arrangement. So it's a cyclic, certainly a cyclical arrangement need to come to that that structure. So that is the next step. So let's see. cyclization. Squalene first undergoes oxidation, and which is the enzyme that is helping the oxidation of squalene? That is epoxidase. Epoxidase helps oxidation, and the hydrogen is received by NADPH, and squalene epoxide is forming there, and still it is a straight chain, and the squalene epoxide undergoes cyclization with the help of the enzyme cyclase, okay, and form the 30 carbon cyclical form. It is called a lanosterol. It is the first sterol formed in this biosynthetic pathway. First sterol. Let's see its structure. So this is the lanosterol. It has more similarity with the cholesterol. Still have some difference. That difference need to adjusted to make it to a cholesterol. Let's see how it is happening. Next, need to cut its size and eliminate, do some modification. So let's see what are those modifications. In the structure of lanosterol, going to remove 3 methyl groups that is located at carbon 4 and carbon 40, and by that way, because of that removal, what is forming? Cymosterol is forming. What is forming? Cymosterol. I will show you. This is lanosterol, and lanosterol in the 4th carbon we can see 2 methyl group, and also in the 14th carbon also 1 methyl group. As these 3 methyl groups are removed, it becomes cymosterol. Then next is some shifting in the position of a double bond that is initially located between 8th and 9th carbon. It is shifting to another position between the 5th and 6th carbon. Because of shifting, what is forming? Desmosterol is forming. Show you. So this is cymosterol, and actually between 8th and 9th, that double bond present, it is shifting to here. So between the 5th and 6th carbon, you can see that double bond. By that way, desmosterol is forming. Okay, and this desmosterol, it is usually present in fetal brain only. If by chance, if it is located in adult brain, that indicates that that person have brain tumor or always gliomas. So it's not good to have this desmosterol in the brain of an adult man. In the fetal, it's not a problem. It's a usual regular thing. Okay. Next, another last modification is happening. That is a reduction to a double bond where it is located between 24th and 25th carbon in the structure of desmosterol. Because of that reduction, cholesterol will form. I will show you that. This is desmosterol, and between the 24th and 25th there is a double bond. It undergoes reduction and form the cholesterol. There is no double bond. So this is the synthetic pathway of cholesterol. I hope that is very clear to you. Okay. Next, we are going to see the regulation of cholesterol synthesis, biosynthesis. So, regulation of cholesterol synthesis. The cholesterol level in our body is regulated by the cholesterol itself. How it is regulating? I already told you that in the cholesterol biosynthetic pathway, the rate limiting step is the reduction of HMG-CoA to form mevalonate, and that enzyme that is helping that reaction is HMG-CoA reductase. So by regulating its action, by regulating its formation, the cholesterol biosynthesis is get regulated in our body. So let's see. So cholesterol itself is regulating the expression of HMG-CoA reductase G to regulate the level of cholesterol in our body. How that regulation happens? First, I am going to talk about the condition when the cholesterol level is low. I am just drawing the picture of that DNA. that is responsible for forming the HMG-CoA reductase enzyme. They present a protein, SREBP protein. It is inactive normally. What's its name? That expansion of that name of that enzyme is sterol regulatory element binding protein. Sterol regulatory element binding protein. It is inactive, and this inactive protein, it go and binds with another protein. Its name is SCAP. SCAP means SREBP protein cleavage activator protein. So this protein has some significance. It is a sensor. It senses the level of cholesterol inside our body. It can sense the low level of cholesterol. So it is the intracellular cholesterol sensor, and it directs that SREBP protein to the Golgi complex. And inside the Golgi complex, certain proteolyses, certain enzymes are present. They are S1P and S2P. It made a cleavage on that SREBP protein and make it active. And as it become active, it can go and bind to this sterol regulatory element that present inside this DNA. As this SREBP protein bind to the sterol regulatory element, this gene become active and it start to transcribe and form the HMG-CoA reductase enzyme. As the HMG-CoA reductase enzyme is formed there, then it is possible to continue the cholesterol biosynthesis. So, its vice versa will happen when the cholesterol level is high. And it is the long-term regulation. I hope it is clear. And also the level of cholesterol can regulate the expression of another gene. Which gene? The gene that is forming, creating the LDL receptor. LDL receptor is a receptor that enables the cholesterol to enter inside normal cells. So that is that receptor. Its gene also. This receptor formation of this receptor also get regulated by the level of cholesterol. Next, we are going to see some short-term regulation. This regulation also focusing or using that main enzyme HMG-CoA reductase through some covalent modification happening to that enzyme. This is our HMG-CoA reductase enzyme. And as it bears a phosphate group, it stay inactive. While it bears a phosphate group, it stay inactive. As this phosphate group is removed, then it becomes active. It can continue the cholesterol biosynthesis. And that removal of phosphate group is done by an enzyme called protein phosphatase 1. Protein phosphatase 1, it goes and it removes that phosphate and make the enzyme active, and it will start to continue the cholesterol biosynthesis when the body have lower level of cholesterol. And in opposite condition, another enzyme AMP activated kinase, it adds the phosphate and make this enzyme inactive when the level of cholesterol is high in our body. So these are the two enzymes that is doing some covalent modification in the enzyme and regulate cholesterol level in body. Other regulations also there, insulin and thyroxine. They are regulating the cholesterol level by how? By increasing the activity of this HMG-CoA. And other hormones also there, cortisol and glucagon. They are decreasing the activity of this enzyme. So that depends upon the level of cholesterol in our body. Certain drugs also there, lovastatin and all other statin groups containing drugs. They are all competitive inhibitors of this enzyme HMG-CoA reductase. By that way, it block the action of this enzyme and prevent the further process of cholesterol biosynthesis, and by that way can cut down or reduce or lower the level of cholesterol inside our body and can prevent any spells of heart attack or cardiac arrest. So that's very, very, very important. I hope that the entire process is very clear to you. I'm going to wind up the class. Thank you.
I should fuck her in the Benz.